The completion of the Human Genome Project in 2003 paved the way for work to get underway in other branches of research. For the first time, an almost complete catalogue of all our genes was now available. Around 20,000 genes provide the blueprints for proteins. However, these blueprints often allow several variants to be drawn up. It's estimated that every human being has a total of some 83,000 different proteins. Taken as a whole, these proteins are referred to as the proteome. Proteins are involved in all the main functions of life. In most cases, they need to act in concert with other proteins. This is also true of some of the most common proteins, that is, the enzymes involved in our metabolism. They sometimes form extensive complexes in order to make better use of nutrients. Different proteins also work together in the muscles, like the individual component parts of an engine. And whenever an external stimulus strikes a cell, a chain of proteins transmits the signal to the cell interior. Proteins resemble a social network in the way they work together. Sometimes the connection is very close and long-lasting, but most of the time it's limited to a brief contact. Some estimates suggest that some 500,000 contacts or interactions are possible between proteins. The entire network of contacts is called the Interactome. Research into the proteome and the interactome provides important clues about the processes that occur inside our cells and organs. It also helps to gain a better understanding of certain diseases. For example, by analyzing the interactome, researchers have shown that in asthma, a certain signaling protein influences the effect of drugs. With some biliary diseases, the proteome can facilitate the diagnosis. That is, when it comes to determining whether a rare form of cancer is involved or a benign growth. And in the case of congenital heart disease, the proteome has provided a starting point for facilitating the treatment offered to many patients. Research is progressing apace, but still has a long way to go. And while it is true that an attendant protein has now been found for more than 90% of genes, the interactome still presents a huge gap. Indeed, only about 10% of the presumed contacts have so far actually been observed. In this respect, current scientific methods are being pushed to their limits. The interactions between proteins are often very short and fleeting, making them all the more difficult to study.